Thanks, Dr. David. I now request Dr. Tony to come and just address us and explain a bit more about the institution that we are meeting in, St. John's Research Institute. Shortly after that, we've very fortunate to have heads of 3 divisions of the institute here with us to explain some of their work, which is very relevant to PORTENT, and we hope it fosters some collaboration as well.
Good morning, everyone. So again, I just thought I'll quickly take you through the history of St. John's. This is like breeze through to give you an idea of what we do at the academy, and then more specifically at the Research Institute. And I'll also give you a brief overview of the division I lead here, the Division of Medical Informatics, and a new entity we created. I'd say not too new, but an entity that we created for fostering and incubating innovations in St. John's, called St. John's Health Innovation Foundation. So that's roughly where you are sitting. We're on a 140-acre campus in South Bangalore. In what you see here, and that's the medical school, that's our hospital, and we have a nursing school, and we still have some greenery on campus, and I hope you get to walk around later today or in the next 2 days. The story of St. John's began way back in 1920 with this young lady you see here. She was Mary Glavery, Dr. Mary Glavery, who finished her medicine at the University of Melbourne. And this was as early as 1914 when she got this calling, and there was a pamphlet that she found on her clinic saying, women and children are dying in India and we need doctors to help. World War I was ongoing, and she waited for the war to settle down, and then boarded a ship and landed in Chennai, Madras that time, and then decided to become a nun. In the old days, you couldn't be a doctor and a nun; you had to, get permission from the Pope. So she wrote to the Pope and said, "I'd like to become a religious sister." And once she landed in Madras, and she was taken to another tiny place called Guntur in our neighboring state, where she started working in the trenches. And she really encountered this whole disparity of health. And that's what motivated her to start nudging the clergy of the time, that's the bishops and the local clergy, to create an institution that will train doctors who will finally go out to the hinterland of the country and serve. Mary Glavery never lived to see her dream. She died in 1957 in Bangalore, and her vision and mission continues to live in St. John's. The CBCI, the Catholic Bishops' Conference of India, then got the permission of the Pope. It chose Bangalore by chance. I'm not sure how they decided that. The first institute that came up was the medical school. We started off in an orphanage in East Bangalore for the first at least 5 years at least, till this building that you see in the forefront. That building was set up with some funding from a German organization. And it was much later, in 1975, you see the hospital behind. It was set up actually by funds from the US government. There was some funds left behind in India from aid, from US aid in the old days, and that was used to build the hospital. A senator called John McCormack was responsible for ensuring that those funds were utilized to build this hospital. So there's a OPD outpatient block called the John McCormack Center at St. John's. Today, the hospital has grown to be a 1,200 bed facility. Almost 2,000 bed facility, and because with operational beds about 1,800 at any point of time. The nursing college was set up in 1918, and that's a very busy nursing school. Institute of Allied Health much later. The Research Institute was then instituted with some funding from one of our alumnus, who's based in McMaster University. Dr. Salim Yusuf was an authority in cardiology. He was nudging the management to tease out research as a separate umbrella institute within the organization, and that's what happened in 2004. And more recently, St. John's is expanding its footprint to other areas of Bangalore. We've now set up 108 secondary hospital in South Bangalore to cater to villages in South Bangalore. And of course, very recently, that's this year, we've created a new entity called the St. John's Geriatric Center to provide long-term care and chronic care for and rehabilitative care for the elderly population of our city. So this is what we look like today, from that dream of Mary Glovery to, what we are today in these various institutions. And this afternoon, we will all be visiting this community center, the Bengaluru Community Center, which is the center where we orient our medical students, nursing students, and other students to rural health. Because Bengaluru used to be a tiny village. It's now grown into a small town, so you will not see it like a village, but you will pass through villages on the way, and you get a glimpse of what rural, Bangalore or rural Karnataka is now. The Research Institute, as I mentioned, was set up in 2004 with this vision and mission to really address these disorders, diseases, infectious diseases, cancer, but through a unique blend of basic science research, clinical research, and population research initiatives. And that's what we've been doing over these 20-odd years. As I mentioned, it's an umbrella institute for the whole academy. All research that happens within St. John's, whether it's in hospital or in medical school or nursing school, is managed by the Research Institute and administered by the Research Institute. But within the Research Institute, over the years, we've created a few focus areas, and we're still evolving and creating new focus areas. So as we started off, our focus was on population health. So we have a division of epidemiology and population health, so maternal and child health. And this is very public health facing, a unit that does a lot of work on public health. We just heard Mary, but she heads division of infectious diseases, and we've been happy to involve her division with the PORTENT malaria testing very recently, and she'll tell you more about her division later. We have a division of nutrition, and here from Rebecca, we'll talk to you about this division as well. Division of clinical research and training. This is a division that does very large scale trials, subjects, over 10,000 subjects at a time, but looking at cardiovascular health primarily, or cerebral vascular health, and looking at interventions, low cost interventions that work in an emergency setting. Now, I'll talk to you more about the medical division of medical informatics. We have a division of mental health and neurosciences that focus again. A lot of it is public health, public mental health. How we have strategized and interventions for mental health in rural settings and primary care settings. Dr. Jyothi will talk to you more about the division of molecular medicine and the work that's happening there. And of course, we have a unique division of health and humanities, which is, which looks at the other aspects of social determinants, ethics, now AI and ethics, and how do we deal with technology, and how do we still maintain our humanity? Certain new areas of work that we're entering into is under Dr. Jyothi's division, we have a hematology research unit. And under this unit, it focuses on looking at, can we work towards gene therapy and for rare diseases like hemophilia, or even looking at sickle cell anemia, or low-cost CAR-T cell therapy. And we have a scientist who's just joined us from another institute who's leading this research. We hope to move into stem cell and regenerative, and these are aspirational areas of work that we'd like to move into. This is an area we still haven't jumped into, climate change and planetary health, but there are a few faculty are very keen, and we've applied for a few grants. I'm hoping to move that forward as well. We have a strong immunology unit, but vaccine research and trials is something that we, again, there's an aspiration to build towards that. And being in India, we have traditional practice of medicine. And there's a new field that we call integrative health, which looks at health holistically, looking at Ayurveda, looking at all the traditional forms of health. And we are hoping to move in that direction as well. As I mentioned, we have a very strong clinical immunology unit led by joint faculty in King's College, and works under the division of infectious diseases under Dr. Mary, and that's something that we've set up. Autism has been a special interest for St. John's, and over the last 5 to 6 years, we've set up a separate center focused on autism research. It's called the Center for Advanced Research on Autism and Developmental Disorders, and they've been doing a phenomenal job. We've got a cohort of about 3,000 patients and looking at autism from multidisciplinary approach, from molecular approach, from clinical approach, computer, computational science and AI. And that's been a very interesting direction that we're taking. And more recently, we are working towards trying to understand how we age healthily, and we've set up as part of the Geriatric Center to the Center for Advanced Research in Geriatrics, aging and geriatrics, and that group has already started a cohort looking at healthy aging. And of course, there's an interest in what might be done as well. And we working with partners, and some of it is already with Saurabh and team, but yes, we already are looking at local partners as well. A quick word about the Division of Medical Informatics. We, focus at using technologies and trade solutions that can, find improved research or education. And a lot of our work started off with creating content for dissemination, that's simple health content. And we did that for a long time, working with the Dash Group. And of course, we helped design the National Health Portal for the Government of India in 2010. We were one of the teams that helped architect the entire National Health Portal, which went live in 2014. Well, more importantly, we are informaticists, so we are looking at the pipeline. How do we ensure that we create the data pipelines and utilize this data to improve our companies? And over the years, we've moved from health literacy and education, medical gaming, developing EHRs and decision support systems. Now, of course, with point of care medical devices, we'll work with various groups, including the PORTENT group. Public health informatics is still a very strong thing to really solve problems in public health. Care coordination and telemedicine is something that we recently started working on, and we're still moving in that direction. Of course, with natural language processing, AI and ML we've also started dabbling with that. And designing for digital health. Design research for digital health, as was mentioned, we're really keen to design the right solutions, not just create a solution that is there, but really scale it up. And research informatics and training is the other activities we do as a bunch. So that's a real quick brief. Some of the work that we've done in primary care is trying to use open source solutions and stitched with all ecosystem in difficult situations. So we've worked trying to use some of these solutions in very remote areas in Karnataka. We also have worked with the University of Washington. This was in 21 round where we looked at developing a mental health app for workers, women primarily who work in little groups in their villages. Many of them are technology challenged, but it was trying to design an app for them and with their participatory design inputs. More recently, we've been working with the Department of Microbiology and another PI to develop an app for antimicrobial stewardship. This is what some documents have been working on. Again, starting with designing the app from the requirements, working with every stakeholder and building up these apps. This is from the Government of India. We were asked to evaluate a very large platform that the Government of India rolled out to study non-communicable diseases, and they were having some challenges. So our team went and did basically understanding what was going on in the field and optimizing this whole application at scale. So this is what we did with some of the collaborators from the Harvard School of Public Health. We were able to provide these inputs. More recently, we've just got a grant. We've built an ultrasound device with our neighboring institute, the University of Science, with a grant from the UK government many years ago. It was in 2020. This prototype was available. Ultrasound is kind of banned in India. You can't develop a portable ultrasound and take it to the field because it's an act that prevents it because of certain gender-based issues. So we've developed a blind ultrasound device which doesn't need sophisticated equipment in the field. We've recently got a grant with from the Indian government to take this prototype from its TRL 3 4 to the next level and validate it in settings. So this is what we've just started developing with. So this is just a summary of what we do. And of course, with AI, we've got our finger in many of these little pies, but still trying to move this whole AI piece. For us, data comes first. And before we fix that, we don't want to jump into AI too aggressively, but we've already started doing it. A quick word about the Health Innovation Foundation. In 2018, the Academy felt that while research was ongoing, they realized that we didn't have the ecosystem. We are a charitable society. We couldn't do anything for profit. So this was discussed with our governing board and leadership, and we then created a company called St. John's Health Innovation Foundation with a focus on health innovation for society. It's a Section 8 company, which means it's a not-for-profit company registered in the Ministry of Corporate Affairs. It's a focus in all these areas: low-cost medical devices, biotechnology, nutrition and well-being, digital health, again, skill development, health literacy. And this is the entity that incubates startups. We have several startups who incubate with us, or we facilitate startups validations through our experts. There are startup who came with a solution to find the best route into the brain for neurosurgeons using AI. So we help facilitate them too. And we've done those kind of validations with actual, with IC approvals, and we facilitate the whole research part. So that's our ecosystem where we have the foundation out here, St. John's out there, and the external environment for funding sources and experts and mentors, etc. So this is also part of the PORTENT group that we're engaged with. In summary, St. John's has been working with the entire life cycle of humans, and we have various cohorts that we work with, and we're very pleased to have you all as collaborators, and we've been engaging with each one of us. So very happy to come to chat later. Thank you.
Thank you, Dr. Tony. We're going to move into a series of introductory talks by some of the division heads. So I'd like to request Dr. Mary Dias to come and present the Division of Infectious Diseases of SJRI. Just a quick request, we're going to try and finish by around 11, so just 10 minutes. Thanks, people.
Thank you. And good morning once again. So I'll just run you briefly through what my division does. Just bird's eye view. But if anybody is interested in looking at more data and other things, the division is always available. So this is our team. Works, it's quite a mobile population. We have a lot of research associates who come and go. So these are our faculty. What I just wanted to stress here is that we have quite a number of adjunct faculty. So people who are interested in working in infectious diseases and have some kind of thing to collaborate with us, we'd always be happy to welcome anyone who wants to work in those positions of adjunct faculty. And we've had a lot of collaborations with these kind of faculty, and we've got good research output there. So to briefly run you through what kind of research we have done and what kind of impact it has had. So some focus areas that we have worked over the past few years. Dengue was a huge problem in 2013 onwards. So it was a felt need to identify dengue fast and to know when to triage patients and to understand who required medical admission and care quicker than the others. So we went in for looking at potential of identifying biomarkers to know, rather than looking at who has got dengue, because that doesn't really make a difference in terms of management. So we could find some potential markers, which we have brought out as publications, but not to really make that kind of impact as of now. So the second thing that we have involved ourselves with the dengue sphere is the validation of tests, which I'm really interested in. That is what is my core interest. So molecular as well as serological diagnostic assays to make them better, quicker, and more accessible to the public. And we've had good collaborations with both semi and governmental agencies towards this kind of work. Then we've also looked at the virology of the whole organism. We have kind of sequenced about 1,000 dengue viral isolates during the epidemics, during the different epidemics, to see if we could identify any kind of vaccine candidates. And we do have vaccine candidate potential, I would say, and we brought it out as a publication. The next area of research that we've looked at for the past so many years, right from the inception of the infectious disease unit, is definitely tuberculosis, which is a huge challenge in India even now. So we've looked at different spheres and aspects of tuberculosis, starting again with diagnostics to look for biomarker signatures, especially to look for treatment response, and also to develop alternate platforms, low-cost platforms to make diagnosis more accessible to the most needy, using different kinds of systems, flow cytometry, microfluidics. We've had a lot of collaborations with the startup industries to see if this could be scalable. We've looked at immunology of tuberculosis, especially BCG. There's a lot of interest now in revaccination and also to look whether this can really be protective. And we found very interesting data. And in fact, actually, we set out a project to study whether BCG is protective and COVID hit us. And so we kind of reappropriated the study and we looked whether BCG could be protective in COVID. And we found quite a bit of interesting data. And so we attributed that to trained immunity. What is, when you see immunity towards tuberculosis protecting against COVID. So these have also come out as publications, which is very, very interesting. And we are still doing the revaccination study, and we're part of many studies which the government is also taking up because this has become a kind of, the next thing, whether TB can be prevented using revaccination using BCG. We've, as a training institute, we've also involved ourselves with training for diagnosis of TB, implementation of the government guidelines. So we've been developing modules in association with the medical education unit of the medical college and to integrate diagnostics with actual implementation of these things. Then recently we've got into clinical trials also, where we have got interest both from the government as well as from private agencies to reappropriate already available drugs and vaccines to prevent tuberculosis. Immunology, as Dr. Tony said, is a new division under the infectious disease unit. And we have a very strong faculty from the King's College London, Dr. Annapurna, who has done a lot of work on immunology of tuberculosis. But after she has come here, we have networked to do other diseases too, including influenza and COVID. And we have two huge projects which are concurrently working on both these important diseases. The other things that we do because of the advent of COVID is surveillance. We have kind of moved into COVID surveillance for wastewaters to predict the outbreak as well as looking at variants. And also looking at antimicrobial resistance, whether in the same vein we could look at antimicrobial resistance, emergence, as well as changing patterns. This is all we are doing in sewage waters. And also we have recently moved into cancer screening, where we are looking for affordable and large scalable diagnostics for diagnosis of HPV in the community. So we have now a good cohort and a place where we are actually doing screening, using, whatever is available, but this is a good cohort to institute and look at any other potential diagnostic tests which can come to screen this large number of people. Other areas that we have worked and working towards HIV, not currently, we are not doing much, but we did have looking at HIV drug-resistant mutations and biological outcomes in people on treatment. And of course, malaria is quite an important thing for us. We have done surveillance for looking at AMR markers in malaria quite some time. Now with the PORTENT, we are looking at point of care test to look at malaria. And we've also looked at AMR and other spheres also, in both diagnostics as well as looking at new classes of antibiotics, which are emerging. One of the ones which came out from our division, although it was not directly under me, but with the collaboration with a startup company, there was a new molecule found, and they are currently evaluating that. So in training, we have a lot of observerships, PhD candidates who come and train with us short term, long term. So this is another potential collaboration. And the kind of facilities that we have at the unit, we have a PSL 3 facility, which allows us to handle infectious agents. So a lot of TB work happens because of this. And also COVID work initially, which was quite an enigma. We used to handle that, but now, of course, we can handle it in BSL too. We have dedicated cell culture rooms, biosafety level 2 laboratories, and we also have a good biorepository where we store all kinds of samples, be it infectious disease or non-infectious diseases, whatever is the projects which are there for long-term storage of the different kinds of samples which are collected during the projects are archived and can be taken on as assignments. And recently, we've also acquired 2 Oxford nanopores, and we are doing a lot of work on sequencing of all the COVID isolates, and we're trying to look at dengue as well as TB, and we are working with another group who's interested. So the novel addition is the human immunology lab and the flow cytometry. So we have a PD-FAX ARIA fusion site flow cytometry, which is capable of single cell sorting. And I'm not an expert on this, I'm speaking on behalf of there. So they do a lot of mRNA sequencing of all the cells which are stimulated by the various antigens, especially vaccine candidates and the new variants of COVID. And they've got very, very interesting work. So our funders, basically both national and international, we're happy to have collaborated with them, and we've got very pleasant interactions with all of them. And we built quite a number of networks, both government as well as non-governmental, and we build trust to work with these people. One important person that I would like to acknowledge here is Sister Aquinas, who's part of Sister Doctors Forum of India. We saw how our network is across the country and was made by sisters. So she's the one whom we actually collaborated with this for protein. Even for the previous malaria surveillance, AMR surveillance that we did, was with Sister, a very enthusiastic sister who was so interested in making accessible healthcare to her poorest tribal area. So with that, I'll just take you through all the other activities that we do. We do a little bit of diagnostic too for the medical school and the hospital. So we have, what I'm trying to say is we have sample pools where we could work on different projects because these are archived. And so we'd like to capacity build, expand infectious disease, especially the genomic sequencing, using that for affordable diagnostics so that we can help the patient faster. Diagnostic stewardship becoming so important these days, and also in training. If there's anything that you want to discuss in depth, we could always do it. Thank you so much.
Thank you, Dr. Mary. Request Dr. Jyothi Prabhu to come and introduce the division in molecular medicine.
Good morning, everyone, and thanks for this opportunity to be included into the PORTENT network. I represent the Division of Molecular Medicine at the St. John's Research Institute. And here we have 4 different units. The first one being the molecular oncology, which is represented by me. Then we have the unit of renal genetics, which is headed by a pediatric nephrologist, a clinician, who's always been interested in looking at developmental disorders in pediatric population related to the kidney, and not only understanding the pathophysiology of this disease, but also the genetics behind it. Then we have a cell biology unit headed by Neha Willis, And more recently, we have a molecular cancer research unit by another young faculty member here. I'll briefly talk about each one of us. So the molecular oncology unit has been the oldest in molecular medicine, almost 17 to 18 years now, who's been exploring breast cancer research. And in this, though we have done multiple studies over the past many years, I would like to focus in the interest of time only on these four main areas which may be of interest to the PORTENT team. So coming to the breast cancer cohorts and the biological specimens. So our focus on breast cancer research has been through building of the cohorts. The first cohort was built from the year 2008 and followed up till 2017, a 10-year-long follow-up, during which time we collected most of the clinical data along with the therapeutic outcome in all of them. And the second cohort started in a decade later, 2019 onwards, and followed up for the next 5 years in a similar fashion. Most of these breast cancer patients were recruited at the time of diagnosis with collection of biological material in the form of blood, frozen samples, as well as formalin-fixed tissue material, which are all safely biobanked, which was used for most of the studies for our molecular analysis in the past decade. Now, as you all know that in the past 2 decades, understanding any diseases has been through multi-omic analysis. But what is actually the drawback of this technology has been that we grind everything and we lose the context of how tissues or cells are organized and how they're related to each other. And in the past few years, technology which has come up is the spatial biology, where we understand, especially in cancer, tumor microenvironment by studying the relationship between different cells as they are sitting in situ or as they are related. And we have recently started exploring tumor microenvironment using one of the platforms. Multiple platforms exist. We are actually trying many of them. So in the past, what we have tried to understand is not only how Indian breast cancer is different, if at all, and how is it different and what are the tumor microenvironments. And this is one of the latest paper which came out recently where we were able to show using spatial profiling in an aggressive subtype of breast cancer called the triple negative breast cancer, where the tumor microenvironment in young breast cancer patients is quite different from that in the older ones, and how that probably has an implications on changing or having a different kind of therapy in younger women. Also to mention here specifically in the breast cancer in India is specifically is somewhat different when compared to the Western cohorts, only because we get more late stage detection, little more aggressive disease, and a higher proportion of younger patients. This is all done by a PhD student and the postdocs named here, Nijesh and Vidya. Moving on, of late, what we have also been doing is a lot of digital pathology. As you all know, it is involving high-resolution slide scanners, image analysis, AI algorithm to quantitatively assess morphology, cellular features, and many more biomarker identification and quantitation. This is all using a single slide, which is just hematoxylin and eosin stained of the histology specimen that we actually get from all our patients. And what we've been able to do is quantitatively assess morphological parameters as well as individual cell types. This is all done with external collaboration with a collaborator, Dr. Namita Shah, who's trained bioengineer, also interested deeply in AI-based algorithms to know morphological patterns in tumors. As I also work extensively with collaborators of pathologists, one of who was very interested to develop these things, Dr. Hafsa. And using both of their inputs, what we have been able to do is create an AI-based algorithm, which is nothing but a predictive risk score, which has stratified the cohorts that I earlier mentioned into risk categories. And you can see that it was able to identify a set of cancers which are likely to do poorly. And we could validate this because of the extensive follow-up that we had curated from all our patients who were built during the cohorts. And the same predictive algorithm was able to perform with the equal sensitivity in the cohort 2, which is an indirect validation of the prediction tools that have been built using this kind of digital pathology platforms. This is an ongoing work. We are looking for collaborators. We want to expand this. As you know, our sample size is small but deeply characterized, and that's how it has helped us to take this to this level, which is showing that deep learning-based prognostic models can be scaled, they are reproducible, and probably they are an interpretation of histological data. Another thing that is of late have been interest for us is development of a liquid biopsy tool. Liquid biopsy, as you all know, is using of a small amount of blood sample to detect any disease, or in case of cancer, to either screen cancer, detect it early, monitor the disease, or find out therapeutic resistance during therapy. And the platforms that exist in India as of now have been most often been much above the levels that our patients can afford, be it the PET scan or of late the relatively new tests such as circulating tumor detections, which cost nothing less than a few lakh of rupees, which is always exceeding the cost of treatment itself, which the patient necessarily deserves to take. And towards this, they were interested in developing a more affordable test. So what we did was trying to identify circulating tumor DNA using a small amount of blood by just developing a small test using, which is a PCR-based platform. The hypothesis here is very simple, that cancers undergo extensive necrosis and hence shed a longer fragment of DNA, whereas the routine apoptotic process in normal individuals should shed a smaller fragment of DNA. And if we manage to take a ratio of the two, cancer patients must have a larger ratio when compared to the normal. And this was the initial study which we did, which is still in progress. We are in the process to improve the sensitivity and the specificity of this test. There is also an interest to add more specific tumor secreted protein markers to increase the both again the sensitivity and the specificity of the test at this point. And I'll be more than happy to share more details during our network collaborations. And moving on, I represent also the other divisions, other units within my division. Dr. Neha Vyas, who has been looking at sonic hedgehog, which is a specific protein, which is packaged in something called as exovesicles, and how it mediates intercellular crosstalk both in development as well as in disease. So she has a model where she has isolated two different pools of exovesicles which are secreted during development as well as in disease. One of these pools contains a sonic hedgehog molecule, which is of a larger size, which is called as P450, whereas the other one is the smaller one, which is called as P150. And her group has extensively validated to figure out what is the function of these pools. And it turns out, though the same cell actually produces both the pools, it is the context which matters. And when a cell makes the larger pool, which is P450, it is pushing the cell more towards stem cell differentiation, whereas when it makes a smaller pool, such as P150, it is actually promoting the cells for canonical signaling as well as differentiation. So she has a very specific model system using chick neural explants, And she has used it both in development as well as in oncology, using chronic myeloid leukemia as the case example for her analysis. She also is very interested in developing low-cost assays using gene expression, PCR-based assays. As you know, therapy resistance in chronic myeloid leukemia is a big problem, though the disease can be handled very well if the patient manages to take treatment on a regular basis. And often the development of resistance is managed by looking at copy number variations, which happens to be a molecular diagnostic test, but expensive and cumbersome to do so. And so she, along with collaborations with multiple clinicians as well as statisticians, has developed an algorithm, a simple statistical tool using gene expression assays to identify TKI resistance in chronic myeloid leukemia. We also have a more recently developed unit headed by Dr. Madhumati Nair. And this is just about a year old, and her focus areas has been looking at solid tumor dynamics using circulating tumor cells, circulating tumor DNA, and microRNA circulomics, as well as development of tissue-based biomarkers and in vitro model systems. In the past year, she has managed to develop a very well established research lab, which can do both blood tissue archive, in vitro animal cell culture facility. And she's also developed multiple collaborations, both intramural as well as outside. And I'm sure she has, in the very recent future, she'll be able to also show multiple studies going ahead, which are all listed here. What I have not been able to list in this presentation is what Dr. Tony just mentioned of adjunct faculty who has recently joined our unit, Professor Alok Srivastava from another eminent institute in India, who's been a pioneer in gene therapy and who's actually looking forward to start his laboratory and do phase 2 kind of clinical trials in the near future. In the interest of time, I would like to stop here and take your questions, if any. If not, at the gathering later. Thank you.
Thank you, ma'am. Just like to request Dr. Rebecca to come to the summit.
Good morning. Welcome to St. John's. So the pleasure is on me. Last minute, quickly wind up. Okay. Thank you. I will try my best. So I'm happy to present the Division of Nutrition. We have, along with me, we have professors, associate professors. We have adjunct faculty. Julia is one of them. We have Dr. Mario Suarez, who is the editor-in-chief of the European Journal of Clinical Nutrition. He's also an adjunct faculty. Along with them, we have more than 150 research staff, 20 PhD students in our division. So our main vision is to translate evidence-based research into practice. And our core areas are, of course, research. We do a lot of training, and you can see that students are sitting in the same hall that you're sitting. We do a lot of community service because nutrition is a lot of community service. And also we have an outpatient department where we see clinical nutrition. And our main core focus areas, we try to look at nutrition across the life cycle, mainly looking at macro and micronutrient deficiencies, non-communicable diseases as a whole. We have cohorts, so I have school cohorts where we have over 10,000 school children and we have followed them up 3 times. We are also currently following up babies from birth to the 3 years of age because we are trying to look at their adiposity. And if adiposity is the reason for metabolic diseases, we look at body composition. We have a very strong body composition laboratory. We are very interested in the first 1,000 days, which is pregnancy and the first 2 years of life. We do studies on elderly and we have set up requirements for them.
And we are moving slowly into environment and adverse health effects. And we also have a faculty who is looking at cost-effective energy and nutrient-dense food products for severe acute malnutrition. Our collaborators, both nationally and internationally, I've listed there. You'll see Cornell in the center of them. And we also have a lot of national collaborators. We work with University of Sydney. We are, I'm not going to name them. We have all of them. We work very closely with them. In fact, the Division of Nutrition was started even before the Research Institute. We moved in to the Research Institute from them. So in 1985, we started. And we have a lot of facilities. Like I said, body composition is one of our main areas. So we have something that you will not, we have something also built a whole body potassium counter here. That is which can measure metabolically active tissue. We have different instruments for body composition. We have the stable isotopic techniques and the core biochemistry lab. Along with Cornell and CDC, we have set up the gold standard method of folate assessment. We have metabolic research where we may do a lot of energy expenditure related studies. And we have a fully equipped molecular and cell biology lab. What I want to also mention here that we are the only collaborating center for the International Atomic Energy Agency, which is a UN agency. So while you hear the term atomic, they do a lot of stable isotopic nutrition research across the life cycle to measure both nutrient requirements, body composition, and several others. So we work with them very closely, and we are the only, as I mentioned, the only collaborating center for nutrition for them. So we don't get a lot of money from them, but we do a lot of training for them, capacity building for them, and we also are part of some of the projects. So across the life cycle, we do have set up requirements using stable isotopes. As I said, we will do a lot of training. Currently, we have 20 PhD students. We also have contributed, besides doing active research, to several of the policies of the country for both energy and protein requirements, for the FAO, for the amino acid requirements. And also, we have looked at a lot of infant and young child feeding guidelines were set up by her. This is a program that is coming next year. Nurture is a program that is coming up next week for 2 weeks. This is Dr. Saurabh, Dr. Julia, all of them are faculty in this program. And last year we started this following on a tradition of called the Bangalore-Boston Nutrition Collaboration, which is where I first met Julia, when we set up the first course together. That was with Harvard, Tufts, and St. John's. And now with COVID, we had stopped that, and now we have moved to a different angle, and we are collaborating. We have moved far, moved even larger. And last year we have over 70 participants. This year we are looking at 100 participants across India, about 8 from Sri Lanka. And we are starting on 21st and we'll be going up to August 1st. We'll have faculty from all across the country, looking at important priority areas. We have the key government people also this coming over and speaking to us. Our future is we want to be good, in a center for excellence for mentoring, training, capacity building. And we also want to build a world-class facility based on research, precision nutrition. We have been talking to Dr. Mehta and trying to be open. We look at both sides of malnutrition, both under and over, and we want to look at various components. We have independently started looking. One of my students looks at gut microbiome in early life from birth to the first year of life. We are hoping to move on to different other areas. And we are nutrition, and thereby there is a lot of hub about wellness in the country, and I know I'm sure globally. So we are hoping to also use our research, translate it into clinical practice, and look at setting up maybe a wellness center. Thank you.